All right, let's get right into it. In this first part of our explainer, we're going to unravel the really fascinating and honestly pretty terrifying link between a super common foodborne bacteria and a very rare life-threatening neurological disorder. It's an incredible story of mistaken identity, all happening at the molecular level, with some absolutely catastrophic consequences.
It all boils down to this one central mystery, a question that just seems to defy all logic. How on earth can a simple case of food poisoning, something most of us have probably had at some point, lead to complete paralysis? To figure that out, we've got to first understand the frightening end result of this chain reaction.
So we're talking about a condition called Guillain-Barré syndrome, or GPS for short. It's this rapidly developing illness where your own immune system basically goes rogue and starts attacking your nerves. And as you can imagine, it can be absolutely devastating.
It often starts out so subtly. A person might just notice a little tingling, kind of a pins and needles feeling, and this weird weakness in their legs. It's the kind of thing you might just brush off at first, but it definitely does not stay in the legs for long.
And this is the really classic and terrifying hallmark of GBS. The weakness ascends. It literally climbs up the body, from the legs to the arms, up the trunk, and in the most severe cases, it paralyzes the muscles you need to breathe. Patients often need a ventilator just to stay alive. This is a true medical emergency, no question.
So the big question is, what triggers this catastrophic autoimmune attack on the nervous system? Now, while a few different infections can come before GBS, there is one prime suspect that stands way, way above all the others. This number is just staggering. In about a third of all GBS cases around the world, the patient will report having had a diarrheal illness, a stomach bug, about 1 to 3 weeks before the paralysis started. And the organism behind that illness? Overwhelmingly, it's Campylobacter jejuni. It is the single most common infectious trigger for GBS, period.
And this really gets to the heart of the paradox here. Campylobacter jejuni isn't some rare, exotic microbe you've never heard of. It is the #1 bacterial cause of food poisoning in the developed world, and we usually get it from undercooked chicken. So you have this incredibly common bug causing an exceptionally rare and severe complication. To understand how that's even possible, we need to take a closer look at the culprit itself.
All right, let's put this bacterium under the microscope. Its unique biology is really the key to understanding both its success as a pathogen and, of course, its role in triggering GBS. So what makes Campylobacter so special? Well, it's a gram-negative rod with this really characteristic curved or spiral shape. And when you see two of them together, they form this S shape that scientists literally call a seagull wing morphology. It's also what we call microaerophilic, which just means it needs an environment with low oxygen. And it's thermophilic. It loves heat. It thrives at 42 degrees Celsius, which, not coincidentally, is the body temperature of a chicken, its favorite host. All of this gives it this darting, corkscrew-like movement that lets it just burrow right through the mucus in our intestines.
And this brings us to what I like to call the fragility paradox. See, outside of a host, Campylobacter is incredibly delicate. It's sensitive to oxygen, it can't survive if it dries out, and it's super easy to kill with proper cooking. But once it gets inside your gut, this fragile little organism transforms into a highly motile, invasive pathogen that can cause some pretty severe disease. It's all thanks to its very specialized toolkit of virulence factors. And it's one of those virulence factors that leads us right to the immunological crime scene. Because the link between the gut infection and the paralysis, well, it isn't a direct attack by the bacteria on the nerves. It's actually a tragic case of mistaken identity, carried out by our very own immune system.
The mechanism behind this whole thing is called molecular mimicry. And the crucial idea is this. A molecule on the surface of the invading pathogen looks so similar to a molecule on our own cells that the immune system gets confused. After it's been trained to attack the pathogen, it just can't tell the difference anymore and starts attacking the host as well.
So let's walk through the exact sequence of events. First, you get infected with Campylobacter jejuni, probably from some contaminated food. Your immune system does its job perfectly and creates antibodies that specifically target molecules on the bacterium's outer surface. The infection clears up in a week or so, no problem. But here's the catch. Those highly specific antibodies are now floating around in your bloodstream on patrol, searching for their target. And tragically, they find something that looks identical on the surface of your own nerve cells.
The specific molecule at the heart of this deception is on the bacterium's surface. It's called lipooligosaccharide, or LOS. In certain strains of Campylobacter, the very tip of this LOS molecule is pretty much a perfect structural match for the gangliosides found on the outer surface of human nerve cells. It's like the bacterium is flying a false flag that looks exactly like our own tissue. And this mimicry is incredibly specific, which is fascinating. As you can see here, if the bacterial LOS mimics the GM1 ganglioside on our motor nerves, the antibodies attack those nerves and you get a purely motor paralysis called AMON. If it mimics GD1A, then both motor and sensory nerves get damaged, and if it mimics the GQ1B ganglioside, which is found on our cranial nerves, it causes a very specific type of GBS called Miller-Fisher syndrome, with paralysis of the eye muscles and other distinct symptoms. The specific disguise the bacteria wears dictates the specific disease you get.
So, after all that, what's the verdict in this immunological crime? What's the really crucial takeaway from this whole devastating chain of events? To sum it all up, Guillain-Barré syndrome after a Campylobacter infection is the ultimate betrayal. The very system that's designed to protect us, our own immune response, is tricked by this molecular mimicry into launching a devastating attack against our nervous system. A common infection leads to a rare catastrophe, all because of a case of molecular mistaken identity.
And a final thought here from a senior clinical professor from our source material. Please like and subscribe for more custom medical videos made just for you. Now, moving on to the next segment.
All right, let's continue our tour of great surgical mimics. We're going to dive into another bacterium that is an absolute master of disguise, Yersinia. So let's get right into this fascinating case of mistaken identity.
So picture this. A patient comes into the ER. They've got a fever, a sharp pain in their right lower quadrant, they feel sick to their stomach, and their white count is through the roof. You're thinking, okay, this is a textbook slam dunk case of acute appendicitis. You're ready to call the surgeon. But what if it's not the appendix at all? The symptoms are a perfect one-to-one match, which creates a huge diagnostic puzzle that can send even experienced doctors down the completely wrong path.
So, if the appendix is totally innocent here, what in the world is actually causing this classic clinical picture? That's the big mystery we need to solve. Well, the culprit is this crafty little gram-negative bacterium called Yersinia enterocolitica. Its whole journey, usually starting from something like undercooked pork or contaminated water, all the way to causing this appendicitis-like pain, it all begins by overcoming one of the body's most powerful defenses.
You know what's really amazing about this bug is how it survives the intense acid bath of our stomach. It has a secret weapon, an enzyme called urease. Yersinia pumps the stuff out, and it breaks down urea into ammonia. Now, ammonia is a base, so it creates this little protective, neutralized cloud right around the bacterium. This shield lets it sail right through the stomach, totally unharmed, and into the intestines. It's just a brilliant survival hack.
So, once it's safely in the small intestine, the real plan of attack begins. After surviving the stomach, it sets up shop way down at the end of the small intestine, in the distal ileum. From there, it uses these specialized gateways in the gut lining called M cells to sneak into the lymphoid tissue underneath. These areas, the Peyer's patches, are basically the security hubs for the intestine.
And this invasion, this is the absolute key to the whole story. The bacteria set off a huge inflammatory response, but not in the appendix. No, the inflammation happens in the lymph nodes that drain that part of the intestine. This condition, this intense swelling and inflammation of these specific lymph nodes, is called mesenteric adenitis. That's the real disease we're looking at.
So let's be super clear. The fire is raging right next to the appendix, not inside it. The appendix itself is just an innocent bystander, completely fine.
And this explains the entire magic trick of pseudoappendicitis. Think about it. It's all about location, location, location. Anatomically, where's the terminal ileum and those inflamed lymph nodes? They're in the right lower quadrant, sitting right next door to the appendix. The pain signals are coming from the exact same neighborhood, which makes it almost impossible to tell the difference from true appendicitis based on symptoms alone.
Now, here's where this pathogen gets even more interesting. It has a very unusual and very dangerous appetite. Yersinia is what we call siderophilic, which is just a fancy way of saying it is an iron-loving bacterium. It absolutely thrives in an iron-rich environment.
And listen, this isn't just some quirky biological fact. This has serious life-or-death consequences.
For patients with conditions that cause their bodies to store too much iron, like hemochromatosis or thalassemia, they are at an incredibly high risk for severe systemic Yersinia infections. For Yersinia, these patients are like an all-you-can-eat iron buffet, letting it multiply like crazy and spill into the bloodstream.
So how do we unmask this thing and avoid a totally unnecessary surgery? Well, the key is in the clues. On a special stool culture, it creates these very distinct bullseye colonies. But really, the definitive tool is an abdominal CT scan. The CT will show you the real story. You'll see a thickened terminal ileum, you'll see those enlarged mesenteric lymph nodes, and here's the smoking gun, a completely normal-looking appendix.
The big takeaway here is simple, but it's so critical. Not all pain in that right lower quadrant is appendicitis. Knowing about these mimics, asking about risk factors, and using the right diagnostic tests like a CT scan, that's how you unmask the imposter and prevent a patient from going to the OR for no reason. Here's a quick clinical pearl for you. Please like and subscribe for more custom medical videos made just for you. Now, moving on to the next segment.
And all of this leaves us with a really provocative thought for our clinical practice. In a busy emergency department with so many patients coming in with belly pain, you just have to wonder, how many of those suspected appendicitis cases are actually one of these great mimics, like Yersinia, in disguise?